Hi everyone, welcome to the session. I want to thank the organizers of Discovery on Target for inviting me to talk to you today. The subject at hand is new therapeutic modalities in cellular immunotherapy. And what I want to focus on is targeting multiple antigens simultaneously. How are we going to do this? How are we going to make it simple and practical? And I will discuss this subject in the context of my role as co-founder, president, and CSO of Aleta Biotherapeutics, a private cell therapy company in Natick, Massachusetts, which leads me to my disclosures. The advent of cell therapy has been remarkable when we think about the impact of anti-CD19 CAR-T cell therapeutics on late-line B-cell leukemia and lymphoma. And by late line, I mean those patients for whom there are no other options. And as you'll see, we have about a 50% durable remission rate in these indications. And we're now approaching the 10-year mark, which means that patients are being declared as cured. So we've moved a patient who was about to die into a cure. That's not a sentence that we can use a lot in the context of such refractory indications. And I think it's a success that we need to build on. So CAR-T cells work amazingly well when they can find and bind to tumor antigens, triggering cytotoxicity, serial killing. Each CAR hits multiple targets. Cell proliferation. Each CAR produces progeny that continue the campaign. Those series of activities lead to a CAR-T cell population that is persistent. And we need CAR-T cells to be persistent for at least a month, 2 months, maybe 3 months, in order to ensure durable and long-term protection for the patient from their tumor. In other words, we develop immune memory from the CAR-T cell pool. Now, we started with anti-CD19 CAR-T cells. They're the ones that are approved. They're also rather extraordinary and exemplary as a CAR-T cell. We don't see other CAR-T cells having quite the properties that anti-CD19 CAR-T cells have. This is in part due to the fact that anti-CD19 CAR-T cells can also see B cells, normal B cells, that are CD19 positive as an antigen source. That gives them an endless source of antigen with which to expand and proliferate. We're beginning to understand that interaction of a CAR-T cell with a normal B cell or with a B lymphoma cell is actually productive for the CAR-T cell. The T cell sees that interaction as a cognate interaction immunologically. It's beneficial to the CAR. That's not true of the interaction with solid tumor cells, for example. CARs to CD19 traffic throughout the patient, solid organs, bone marrow, circulation, the central nervous system. Now, these are the best CAR-T cells we have today, but even these are limited in their efficacy, and they're limited by a relapse rate that approaches 50% in most trials. And that relapse rate is driven in part by loss of antigen from the tumor cells, so we see antigen escape. As we turn from the B-cell malignancies to solid tumors, the issue of antigen escape and antigen heterogeneity becomes ever more important and more difficult to overcome. So the problem in moving CAR-T therapy more broadly into other indications is that cell therapeutics will be confronted by the issue of tumor heterogeneity and loss of antigen in response to therapy. So we've got to figure out how to do multi-antigen targeting. This is not a new thought, and there are many ways that people are thinking about doing this, is illustrated in this and many other reviews. So this cartoon here shows us mixtures of CAR-T cells, tandem CARs, which are bispecific CAR engineered dual CAR-T cells, again, CARs that are engineered to have 2 CAR domains expressed on their surface. We also have examples of CARs that can secrete another element like a BiTE. That gives you a couple of antigens per CAR. And then, of course, all of the adapter protein companies that try to use a universal CAR and then link it to antigens by systemically adding an adapter protein. These are all very elegant solutions to the problem of multi-antigen targeting, complex engineering, complex clinical translation, and they solve one problem, one antigen at a time. Of course, one issue is that that's not the only problem confronting cell therapy for solid tumors. We also have to deal with challenging tumor microenvironments and deal with CAR-T cell persistence. It is notable that CARs to solid tumors tend not to persist very long in the patient. So this is from the same review, showing a number of ways that people think about enhancing CAR-T cell persistence, perhaps by choosing different T cell subsets, by providing cytokines or artificial antigen presenting cells to boost the CAR-T cell population. But if we think about this globally, we now have to add these solutions back on top of the multi-antigen solutions. So it involves more engineering and more, I think, challenges in terms of successful clinical translation. So we took a different approach, which is to go back to those anti-CD19 CAR-T cells. And we said to ourselves, well, these CAR-T cells are unique. They have inherent persistence properties because they can see CD19 on normal B cells. And that means, for example, in the context of B cell malignancies that also express CD19, that the CARs can outlast and outlive the tumor cells because they can keep seeing CD19 on B cells long after the tumor cells are gone. That's what gets these patients into MRD negative status and allows them to be cured and have long-term durable remissions. So the idea is to leverage those CARs as a platform, as a new therapeutic platform, because they've got the persistence advantage, because they have a fitness advantage, and because we know how to make them. CARs to CD19 are the ones that are approved. There are many programs worldwide. The CMC challenges, the release assays, etc. are well understood. So how do we leverage this exemplary CAR for our own purposes? And the way we've envisioned doing this is by building bridging proteins that act as anti-CD19 CAR-T cell engager proteins. So that means that they have CD19 as one component of the protein. It's the extracellular domain of CD19, quite small. It's a universal target for every CAR to CD19 on the planet. The other end of the bridging proteins are antigen binding domains derived from antibodies that bind to tumor antigens. These can be SCFEs, they can be VHs from llama, they can be fibronectin type 3 domains. They can bind one antigen, they can bind two antigens, they can bind three antigens, and we can introduce additional functionality. For example, we can bind to an antigen, but we can then also bind to anti-albumin to extend the VH, extend the half-life. So we like bridging proteins. They enable dual and triple antigen targeting readily. They're small. They're quite modular. We can move pieces around. They can be engineered to be expressed by CAR-T cells themselves. They can be engineered to be expressed by oncolytic viral vectors, and we can purify them as biologics for injection. That presupposes that we can tune their PK properties. So as a biologic for injection, we need to have a long half-life. Perhaps if we're secreting it from a CAR-T cell, we want to have a short half-life to limit systemic distribution of the antigen binding component. Modular, multi-antigen, adaptable. This is how they work. It's quite simple. The antigen binding domain binds to the antigen and expresses CD19 on the surface of the target cell. It's shown in a cartoon form here, and we can see it by flow cytometry with an anti-CD19 FACS. So if this is a solid tumor cell, normally CD19 negative, we've now made it CD19 positive, and we can see it by adding the bridging protein. We can see it by staining for CD19. So we've coated the cell with CD19. It follows that if we now add a CAR a CAR-T cell that recognizes CD19. We can kill that coded tumor cell, and indeed we can, in a dose-dependent manner. These are controls down here. These are a couple of bridging proteins to Her2. And the potency is extreme, which is great because that means we don't have to provide a lot of protein in order to induce tumor cell killing. So what are we going to do with this that makes sense clinically? So the first program I want to talk about is a program for the treatment of breast, lung cancer, and perhaps colorectal cancer patients that are Her2 positive and are suffering from CNS metastatic disease. So Her2 positive cancer patients as a class across indications tend to metastasize to the CNS. So there are a lot of patients, as you can see here. Fair percentage of them are Her2 positive. And those that are Her2 positive, when they develop metastatic disease, so they become refractory, they tend to metastasize to the CNS. And that's bad news. They have a poor prognosis and limited therapeutic options. Importantly, the antibodies that are used to treat CNS lesions, those antibodies can't treat CNS lesions because they cannot cross the blood-brain barrier. What can cross the blood-brain barrier are CAR-T cells, as we discussed earlier. So here, the bridging protein is actually engineered to be secreted by the CAR-T cell. So we have an anti-CD19 CAR domain, so that's put on the cell surface. We have a cleavage site, and then we have the bridging protein secreted. So this T cell is secreting a bridging protein that will coat a tumor cell with CD19, enabling that T cell to then kill the target tumor cell that it's just coated with CD19. Works very well, so the secretion is constitutive in the nanogram per mil range, coats tumor cells that express the target antigen avidly with CD19, triggering sub-nanomolar potency killing. So that's good because we don't have to secrete a lot of bridging protein in order to effectively and efficiently eliminate target tumor cells. That's shown in the in vivo model, whereby we've implanted a Her2 positive cell, we've allowed it to grow, and then we've added these CARs to CD19 that are secreting the bridging protein. They find the tumor cell and eliminate it, curing the mice, the controls. By day 40 are all dead. We wanted to go back and forth between native CD19 and coded CD19 in this model. So after we rested the cured mice, we gave them a NOM6 cell that's CD19 positive. So these mice that have already cleared a Her2 positive SCOV3 ovarian carcinoma cell go on to clear the NOM6 CD19 positive cell. They're flatlined here. The naive controls, of course, light up with leukemia very quickly. This just shows we can go back and forth between the antigen expressed on a cell, the antigen painted on a cell, just as we would in the patient. All right, so that is a single antigen targeting bridging protein, CD19 anti-Her2. Where do we go from there? So if we think about primary CNS tumors and think about the antigens that are expressed on primary CNS tumors, we come up with a pattern that looks like this. We have a lot of indications in which Her2 is expressed, a lot of indications in which these other three antigens are expressed, B7H3, H6, and IL-13 receptor alpha 2. So we have a range of indications, we have a range of antigens. What can we do with it? So just to quickly walk through how these campaigns go. Once we've selected a series of antigens, we immunize multiple llamas, we derive libraries, and then we pan those libraries by expressing the sequences on phage. We do panning and derive the llama VHs. These tend to be very, very high binding affinity and therefore very high potency when we build them into a bridging protein format. So this is an example of what a multi-antigen binding bridging protein looks like, and the key components are the CD19 extracellular domain, that's the target for the CAR, and then the two antigen binders. And you'll notice here we've moved Her2 to the N terminus. We've put B7H3VH, the C terminus CD19 in the middle. This bridging protein can bind to either target protein individually or simultaneously. and kill in either case. So if we bind to HER2, add a CAR, we kill. Bind to B7H3, add a CAR, we kill. So it's not a gate, it's either or, and that means we can kill single antigen positive cells very effectively. We can kill dual antigen positive cells even more effectively. That affinity shift is shown here. We're now driving an IC50 that instead of being in low nanograms per mil is in picograms per mil. This is a amount of protein readily secreted from a transduced T cell. So these CNS programs, whether it's the CNS metastatic disease program or the primary CNS programs, are indications where this particular technology provides a unique advantage. We can infuse the CAR-T cells systemically, allow the CAR-T cell population to expand and persist using CD19 positive B cells, traffic into the CNS where locally they'll expand at the site of antigen positive tumors in the CNS. Importantly, for the HER2 positive CNS Mets program, patients can remain on standard of care, which is anti-HER2 therapy, typically trastuzumab, pertuzumab dual, or a trastuzumab-based ADC. They don't have to come off treatment because we don't need the HER2 in the periphery in order for the CAR-T cells to expand. That therapeutic profile is wholly unique to our program and cannot be achieved by CARs directed to HER2 alone or by any of the biologics that can't cross the blood-brain barrier. For the primary CNS tumors, the program is uniquely designed to simultaneously attack multiple antigens with a single CAR, and we're building dual and triple antigen binders now for the use in those indications. Again, systemic administration will be important. We can work in the adjuvant setting, we can work in the neoadjuvant setting, and that means we can work in refractory patients, we can work in frontline patients. For example, in a clinical trial that moved from dose escalation in the refractory setting to expansion in the newly diagnosed setting to achieve maximum benefit for the patient. So this is what our pipeline looks like. I've only talked about the top part today. These are the two programs in lentiviral vectors in development now. This vector is in development now. We have biologics programs built to support existing CAR-T cells. So CARs to CD19, CARs to BCMA, the CD19 biologic program at CD19 anti-CD20, also in development now. And we've got a multi-antigen targeting AML program in the works. So I'll stop there and say thank you very much.